amazing opportunity. Thank you for the amazing opportunity to present here for all of you. It's an honor to be invited. I will now share my screen. And just a heads up that I'm using a wider screen on my monitor is very wide. So on your end, the slides may look a little bit smaller, but there's an option on Zoom in which you can zoom and increase the size of the slides a little bit. So in case it is showing up small, try to do that in case you're not seeing it very well. So thank you for this amazing opportunity. And it is an honor to be presenting the work that we've recently published at Immunity here at Open Box Science. And so in this work, we found evidence that meningeal lymphatic function is relevant for oligodendrocyte survival and brain myelination. So the lymphatic vascular system is composed, let me just switch this. The lymphatic vascular system is composed of the draining lymphatic vessels, lymph nodes, and lymphoid organs, such as the thymus and the spleen, for example. And its main function is to transport interstitial fluid, macromolecules, immune cells to the lymph nodes, and then from these back into the blood circulation. Thus, the lymphatic system participates in the regulation of tissue fluid homeostasis and in immune cell trafficking. And we also know that the lymphatic system is involved in absorption of dietary fats through these lymphatic capillaries that are located in the intestine villi called lacteals, and in the removal of cholesterol from peripheral tissues to the bloodstream via reverse cholesterol transport. So the lymphatic system is also involved in transport and distribution of lipids in the body. And up until recently, the central nervous system was considered immune privileged. And one of the reasons for that was the absence of lymphatic vessels and drainage in the central nervous system. However, all of that changed in 2015. There was some hints from previous studies, from very ancient studies actually, but in 2015, bona fide markers of lymphatic endothelial cells were found to be expressed in the dura, which is one of the meningeal layers that surrounds the brain. So lymphatic vessels do not actually penetrate inside the brain parenchyma, but they are located in one of the meningeal layers, the dura. And in this dural hole mount, you can see LIVE1 positive. LIVE1 is one of these bona fide lymphatic vasculature markers. And you can see LIVE1 positive vessels, which are distributed along the superior sagittal and the transverse sinuses. And other markers include, that are also expressed, include PROX1, VEGFR3, CCL21, and protoplanin. And at the functional level, these meningeal lymphatic vessels are constantly draining CSF to cervical lymph nodes. And it's not only solids and molecules that are present in the CSF, but immune cells are also being drained. But unlike most peripheral lymphatic vasculature, which develops embryonically, the lymphatic vessels which are present in the meninges, they only develop postnatally. Here at V8, there are a lot of LIVE1 positive cells, but these are all independent cells. And then vessels start to show up and they become fully mature at around P28. So there are different growth factors which are important for lymphogenesis. And we know that the signaling of VEGF-C through VEGFR3 is necessary for the development of meningeal lymphatic vessels. So in this experiment, when P0 pups were injected with an AAV that expresses the extracellular domain 1 to 4 of VEGFR3, and this will lead to the production of a portion of the VEGFR3 that binds to both VEGF-C and D, and thus it's functioning as a trap. So this product will be in circulation, it will trap VEGF-C and D, and it will prevent it from getting into the meningeal, into the meninges, And so it prevents the formation of meningeal lymphatic vessels. While in control animals, which are injected with an AAV that express the portion 4 to 7 of the VEGFR3 that does not bind to either VEGF-C or D, then the vessels are formed. And similar loss of function studies that were performed now in adult mice have actually found that the maintenance of the meningeal lymphatics in adults are also dependent on a constant signaling from VEGF-C through VEGFR3. So in these experiments that were performed by others and by us in this work, when young adult animals were injected with this AAV that leads to the formation of a VEGF-CD trap, then there is a regression of the meningeal lymphatic vessels, which in our experiments, it was to about an ablation to about half of the proportion of the vessels that were present in the controls. So we have taken advantage of this model to explore this question. Can defective meningeal lymphatic function impact brain lipid composition and axonal myelination? And why would we want to study brain myelination? So I mentioned before that in the periphery, the lymphatic vessels are involved in the absorption of dietary lipids, and lipids represent about 50% of the brain's dry weight. And as you may all know, the majority of these lipids are present in the myelin sheath that surrounds and insulates axons. So this question is highly relevant. So starting with a broader analysis, we performed lipidomic analysis on the forebrain of animals with either intact meningeal lymphatics or animals with ablated meningeal lymphatics. And this, once again, stress out what the model is. This is caused by decreased circulating levels of VEGF-C and D, and it's this VEGF-CD trap group. So we found that all the significantly altered lipid species, they were all decreased compared to controls. Then when we quantified the expression levels of the myelin protein MVP at 7 weeks after the first AAV injection, we found a decreased expression in the VEGF-CD trap group using two techniques, immunofluorescence and Western blots, compared with the control animals. We observed this decrease, of course. And regarding mature oligodendrocytes, which are the main cells expressing MVP and that are responsible for the formation of myelin, we found an overall decrease of the density of mature oligodendrocytes, which were identified by the CC1 marker. And we observed this not only in the corpus callosum, which is a white matter region, but also in the gray matter at the cortical level. So this seems to be a wide decrease in the number of mature oligodendrocytes. So it does seem that impairing meningeal lymphatic function causes demyelination. And so we next wondered what cell-specific gene signatures were being triggered by this loss of meningeal lymphatic drainage, and that could be preceding this loss of mature oligodendrocytes. So in the results that I just showed you, the majority or the more drastic alterations, we were observing them at 7 weeks, a little bit a later time point. So in this experiment, we looked at the 5-week time point. A little bit early of this, there was already a smaller decrease in the number of mature oligodendrocytes, but it wasn't very pronounced. And we collected the forebrain of the animals for single-cell RNA sequencing. And we identified 21 cell clusters. And actually, the ones that presented higher number of differentially expressed genes were the mature oligodendrocyte clusters along with astrocyte clusters. And so we focused our attention in the mature oligodendrocytes. And when we performed pathway analysis on these DEGs, it revealed that mitochondrial metabolic processes, such as oxidative phosphorylation and response to oxidative stress, were altered in the VEGF-CD trap group. And these changes in oxidative stress pathways were further corroborated by a decrease in the antioxidant enzyme superoxide dismutase 2. But the model that we've been using so far, it does present with a big caveat, which is we are impairing VEGF-C and D signaling in the entire body. And we are preventing it from binding to the receptors present in all cell types. So we next generated an inducible model of lymphatic endothelial cell loss. So we crossed CDH5 CRE-ERT2 mice, and CDH5 is an endothelial promoter with animals flocks for PROX1, which is a transcription factor important for the induction of a lymphatic endothelial phenotype. So after injecting tamoxifen in these animals, in young adult animals, we induced CRE recombination, and we confirmed that this model was working since 3 weeks after the first tamoxifen injection, there was a decrease in the coverage of LIVE-1 positive vessels in the meninges. However, in the control animals, which are also CDH5 CRE, but are not PROX1 is not flux, then tamoxifen injections did not affect the coverage of LIVE-1 positive vessels. So this is also good. And then we quantified the density of mature oligodendrocytes. And once again, we recapitulated our previous results, and we found a decreased number of both mature oligodendrocytes in the corpus callosum and in the motor cortex. But what we found more interesting was that there was a correlation between the degree of meningeal lymphatic vessel ablation and the number of mature oligodendrocytes. And then we decided to perform electron microscopy. And we found that indeed there was a decrease in the percentage of myelinated axons in the corpus callosum, indicating more demyelination, but there was no alterations in the total number of axons. We also found an increase in the myelin G ratio, which means that the myelin sheet is thinner in CDH5 CRE PROX1 flux animals compared with controls. And so altogether, all these results further substantiate that proper meningeal lymphatic function is necessary for brain myelination. So an adequate drainage is important for maintaining tolerance towards central nervous system antigens. And several studies previously done have shown that lymphatic drainage has a substantial impact on the central nervous system immune responses. So then our next question was whether these two models of impaired meningeal lymphatic function, if they showed alterations in adaptive and or innate immune cells. And indeed, in both of these models, the VEGF-C/D trap and the genetic model of meningeal lymphatic ablation, they both presented increased recruitment of T cells and B cells to the meninges. And in the brain, the total number of innate IVA1 positive cells was similar between both experimental groups, but the CD68 expression on a per cell basis was higher in CDH5 CRE PROX1 flux animals compared to controls, which indicates that microglia is more activated in these animals. So then we wanted to test if we impair either the innate or the adaptive immune system and then induce, if we have impairments in either of these immune responses, and we induce meningeal lymphatic ablation, do we have, is this going to affect the demyelination that we saw or not? So we started by using the RAG2 knockout animals that do not produce mature T and B cells. And so we use these animals, and in some of them, we injected with the AAV producing the VEGF-C/D trap, and others with the AAV, the control AAV. And now, in these animals, we did not find differences in MBP coverage. And also, there were no differences in the number of mature oligodendrocytes in the brain. Then, to study the involvement of the innate immune system, we fed animals with PLX, 5622, which is a CSF1 receptor inhibitor. And at the dosage that we used, we found an ablation of about half of the IVO1 positive cells in the brain. We used 600 ppm. And in this model, similarly to what we observed in the RAG2 knockouts, there was no differences in MBP coverage in the brain and no differences in mature oligodendrocyte lineage. So altogether, these results suggest that when we have defective meningeal lymphatic drainage, this is linked to the generation of deleterious brain-associated immune responses, which are not favorable to the survival of mature oligodendrocytes and myelin maintenance. So having observed the impact of meningeal lymphatic function on demyelination in adult mice, we then asked if the remyelination processes were also affected. So for that, we used the well-established cuprizone model. And because in this model, it is known that after a shorter period of cuprizone exposure, for example, 4 weeks, if we remove cuprizone from the diet, there is spontaneous remyelination that occurs in the animals. And so we evaluated 2 time points, 2 and 4 weeks of remyelination. And we found that at 2 weeks of remyelination, the animals with impaired meningeal lymphatic function had decreased MBP coverage and decreased numbers of mature oligodendrocytes, but this difference was no longer present at 4 weeks of remyelination. So this suggests that impaired meningeal lymphatic function is delaying remyelination, but is not fully preventing it. And when we performed electron microscopy analysis at the 2-week remyelination time point, we did not find differences in the G ratio or in the total number of myelinated axons, but we did have fewer axons in animals with impaired meningeal lymphatics compared with animals with intact meningeal lymphatics. So to further explore what alterations could be contributing to this delay in remyelination, still at the 2-week remyelination time point, we collected the forebrain of the animals and we performed proteomic analysis. And we found that animals with the impaired meningeal lymphatic function presented an immunosuppressive signature, which was characterized by a decrease in several cytokines and chemokines. So since the cuprizone model is not characterized by peripheral lymphocyte recruitment and infiltration into the brain, then we used once again the RAG2 knockout animals to see if we could rescue, similarly to what we have observed before, to see if we could rescue this delay in remyelination. And indeed, not only was the levels of cytokines and chemokines reestablished to the levels of animals with intact meningeal lymphatics, but also the number of mature oligodendrocytes was rescued to control levels. So altogether, these results using mouse models suggest that impaired meningeal lymphatic function is associated with abnormal immune responses that lead to defective brain myelination and remyelination. So then we moved to the human context, and then we wanted to assess potential alterations in lymphatic-related biomarkers in patients with central nervous system demyelination. And for that, we chose multiple sclerosis patients, and we quantified the VEGF-C levels in the plasma and did not find any differences compared with healthy controls. However, VEGF-C levels in the CSF of MS patients was decreased compared to controls. And what we found even more interesting is that VEGF-C levels were even lower in patients that had a relapse closer to the date of sample collection. So if the patients had had a relapse anytime before 60 days of the CSF collection, their VEGF-C CSF levels were even lower compared with patients that were relapse-free for longer. So I leave with these highlights of this work. meningeal lymphatic vessel ablation causes loss of mature oligodendrocytes. Demyelination due to meningeal lymphatic ablation relies and is dependent on a competent immune system. Meningeal lymphatic dysfunction affects brain remyelination and aggravates axonal loss. And multiple sclerosis patients have decreased VEGF-C levels in the CSF. And all of this work could not have been done without the amazing support of the Damskita lab. A big thank you to Sandro. He was amazing throughout all of this work. And thank you to Shannon and Lupe, Megan, Nicole, Ray, that were the people that helped me so much that contributed the most to this work. Thank you to all our collaborators, to all the people that contributed from within Mayo and institutes abroad. And thank you to all the funding sources. And the floor is now open for any questions.
Thank you, Sophia. That was very clear. And I like the science. They do step by step and try to identify a new phenomenon, and which is contributed by immune cells. So now it's Q&A time. And before that, I would like to thank everyone for joining today's seminar. If you want to host or you want to speak in the future, you can fill out the form. I send it to the chat right now. Okay, so now I will allow everyone to unmute.
Should I leave this slide on, or
oh yeah, they can leave the slide on. Yeah, you may need it later, and you feel free to share your video to interact with Sophia directly. So I think what I'm interested in is you saw increased CD3 T cells in the meninges after ablation, the lymphatics, and you use the RAG knockout to show that the lymphocytes might be important. So do you know which subset exactly? CD4, CD8, B cells?
We haven't been, so in this analysis, we did find I only showed increase in overall CD3 T cells. And when we looked specific, if I remember correctly, CD4 had a more pronounced increase related to CD8, but we didn't delve too much into specific types of populations. What were the populations, or we didn't study that into more depth. The only model that we used was the RAG2 knockouts, which are deficient for both T and B cells. So it could even not be a CD4 or CD8, it could even be a more B cell related thing. So then the next approach is we could use depleting antibodies for specific T and B cell types, and then further explore the importance of each one of these cell subtypes.
And those CD3 T cells, are they infiltrated after you ablating the meningeal lymphatic, or they are proliferated locally?
So this increase in the T cells and in the B cells, it's not within the brain parenchyma. It's in the meninges itself. Yeah. And so T cells are usually already there. Right. Basically, what we see is just an increase. Proliferation, we didn't do any Ki-67 or anything. So we don't know if, we think that it might be recruitment, but we don't know if they are proliferating locally, because we didn't use any Ki-67 or other proliferation marker.
Yeah, yes. And the recent publication identified the meninges associated lymphoid tissue. Do you notice that too, or is that tissue affected after meningeal lymphatic ablation?
We didn't look at it.
Okay, yeah, I think it's very new. It's probably published at the same time when you publish it.
We did. I didn't show that part, but also when we looked at deep cervical lymph nodes, there is also an increase in the number of B and T cells present there. So it's kind of an overall thing, at least in the brain border and where it's draining to. We didn't look, compare with blood, we don't see, for example, differences. So it does also seem like it's a thing more actually associated with the central nervous system, let's say. It's not like a broad, like animals start just overall producing more of these TRP cells. So that's why it looks like it's more maybe a recruitment thing, that they are going a little bit more into these regions, these tissues.
Questions from the audience? They're quiet today. If not, I have another technical. Oh, yeah, shall we?
Yeah, wonderful talk. I have a question. So the question is, the recruitment of these kinds of TB, these adaptive immune cells, whether do you think it is caused by the damage of the brain, leaded by the lymphatic vessel damage, or it is have impact on their interactions, especially during the endothelia on the lymphatic vessels and their interaction with these adaptive immune cells?
So yes, we don't know, because like I was mentioning, even though the drainage is impaired because we have less vessels, there is also an increase in B and T cells within the deep cervical lymph nodes. And so if there's an increase there, that cannot be coming from the drainage. So we don't really know where these cells are coming from. And with the PLX model, for example, which is now innate immune cells, we also kind of see a rescue. So it might just be that because the dura is so important for immune surveillance and tolerance and all of these mechanisms, the fact that now the drainage is not being performed correctly, then immune signals that are damaging are now being produced and everything, and are leading to this loss of oligodendrocyte. But we don't really know where the cells are. We didn't explore too much what types of cells are being recruited, where are they coming from. We haven't gone into detail in terms of that.
Thanks, wonderful talk.
Thank you. Thank you for your question.
Thank you. Any other question? If not, I want to ask you a technical question. So do you have experience with an antibody that recognized defective MVP? Have you heard of that?
I did try an antibody that binds to DMBP.
Right, yeah.
So kind of a demyelinated MBP, but
yeah.
the signal so the images that I show for MBP, which are like those almost whole mounds, an entire tissue, even with the cuprizone, when I did that DMBP, I couldn't have like a good decreased signal compared to controls that I could have like use that as relevant in our case. I don't know if I'm making So when I tested it, I also tested it in cuprizone animals, and I didn't see an obvious decrease in the cuprizone compared to controls with animals with normal diet. So then I just ended up not evaluating it, and that was the only test that I did. We do show it in the paper, also stating for another myelin protein, which is MOG, and in that case, we don't see a significant reduction. So, but this could be, could have many reasons. MVP could just be much more expressed compared with MOG, or their location within the myelin, that some of it is more affected than others or something else. But I didn't test other antibodies. Only that.
So in summary, the DMPP antibody,
it didn't work very well.
Even in the cuprizone model.
Yes, even in the cuprizone. For when I tested it, there were other people in the lab next to us that were using it, and they suggested it to me. But since I didn't see an obvious thing that when I quantified it, because that's almost like a positive control, right? So this was supposed to stain more for not the demyelination, but debris, MBP debris and everything. And since I didn't see like a very distinct signal from these two situations, which the cuprizone should be my positive control, then I ended up not evaluating our meningeal lymphatic ablation because I wouldn't see anything there.
Yeah, good to know. Thank you. Okay. Any other questions? If not, I would like to thank Sofia again, presenting your beautiful talk at Open Box Science seminar today.
Okay.
Thank you
you
so
very
much.
much.
Yeah. Have a good
And
night.
if there's any questions, they can reach out to you and you can forward them to me and we'll talk by email.
We'll do that. Thank you.
Yeah, of course. Thank you for the opportunity. Bye.
Bye bye.
Have a good weekend. It's almost there.
You too.